Hey there, budding biologists! Welcome back to the second part of the roles of enzymes in cells. In this clip, we'll be exploring the effect of the environment on enzyme activity. This clip will prepare you for a series of investigations that we're going to be completing in class. Recall from our previous clip, enzymes are highly specific due to their chemical structure. They are proteins that are folded that will be significantly changed by fluctuations in their environment. The three main factors that we'll be exploring on enzyme activity are pH, temperature, and substrate concentration. Each of these factors will either act on the active site shape or the rate of substrate-enzyme interactions. Due to enzymes being made of proteins, their shape can be significantly altered, and this process is called denaturation. It refers to the loss of folding structure of the enzyme, resulting in the loss of that specific active site. This means that the enzyme can no longer act on the substrate. Denaturation occurs outside the optimal pH and the optimal temperature. This is because of the protein structure of amino acid chains. Their chemical bonds are affected by acidity, alkalinity, and temperature within the environment. So let's review this before we get into each environmental factor in more detail. Enzymes become denatured in environments outside their optimal pH or temperature. This causes a change in protein structure, causing the loss of the active site. This results in the enzyme being unable to act on the substrate. Let's first explore the temperature effect on enzyme activity. In many types of chemical reactions, at low temperatures, the rate of reaction is limited due to the amount of energy in the system. This means that the substrate and the enzyme are not encountering each other as quickly because they are moving slower in lower temperatures. As the temperature increases towards the optimal rate of reaction increases. Past the optimal temperature, the enzymes will denature. The heat is changing the chemical bonds in the amino acid chains, causing the unfolding of the enzyme shape. This causes to a loss of the active site, and the substrate can no longer interact with the enzyme. This then causes the enzyme activity to drop off quickly, as seen in the diagram. So, let's summarize. As temperature increases to the optimal temperature, the enzyme activity increases. Past the optimal temperature, it drops off quickly due to denaturation. The second environmental factor is pH. pH is measured in the amount of hydrogen ions in solution. These charged ions have an effect on the bonds between amino acids, changing the structure of the enzyme. In both acidic and alkaline environments, they can denature enzymes. However, specific types of enzymes are found in different parts of the body. For example, protein digesting enzyme pepsin and renin are found in gastric juices of the stomach. They function best in a strong acid. The enzymes in the salivary amylase found in saliva help break down starch and work best in a weak alkaline medium. The action of amylase on starch stops foods passing to the stomach because of the low pH in the gastric juice. So in short, pH optimums are specific to each enzyme. Outside the pH optimal, enzymes will denature, causing a reduction in enzyme activity. Lastly, substrate concentration is the amount of substrate available to react with the enzyme. Let's explore concentration for a moment. Recall from your prior knowledge in science. The concentration of a solution is measured the amount of solute that has been dissolved in a given amount of solvent or solution. A concentrated solution is one that has a relatively large amount of dissolved solute. A dilute solution has a relatively small amount of dissolved solute. The concentration of enzyme compared to the concentration of substrate affects the rate of reaction. If enzyme concentration is higher than substrate concentration, the reaction will occur over a short period of time. This is because there are many active sites available for the reaction. As substrate concentration increases, the rate of reaction will increase because it allows for more chemical reactions to occur. This will continue to happen until the point at which all the enzymes are engaged in enzyme-substrate complexes. This is called saturation. This will then cause the rate of reaction to plateau. This is because the set amount of enzyme can only perform a set amount of reactions at a set rate. So let's summarize. Enzyme activity increases with the amount of substrate to the point of saturation. That then causes a plateau in enzyme activity. In this clip, we have reviewed 3 environmental conditions that affect the rate of reaction in enzymes. Optimal pH and temperature conditions are required, otherwise the chemical bonds are changed in the protein structure of the enzyme, causing a change to the active site. This is called denaturation. The concentration of substrate determines the rate of reaction up to saturation. It causes an increase in reaction up to saturation, which causes a plateau. Okay, hope to see you next time, budding biologists.